Click for IB Biology. Here we're looking at how conditions within the cell, such as temperature and pH, may affect enzyme function. There's a PDF of this presentation in the description down below and a quiz that you can take to check your understanding. As we move into this work, it's important to remember that enzymes are proteins, and they are proteins with complex globular shape. And if that shape, and that particular shape, determines the function. So in the case of an enzyme molecule, the shape issue really is to do with the active site. Therefore, if there are factors that change the shape, change the protein molecule and change the shape, then it is likely that the active site will also be changed, and this may alter the function. More likely, it will lead to a loss of function. So change of shape may result in a loss of function. Looking at temperature, the effect of temperature on enzyme catalyzed reactions. In yellow with the red bounding line is the classic curve of the effect of temperature on the rate of an enzyme catalyzed reaction. However, this is actually a consequence of two separate ideas. A, which corresponds to this graph here, relates to the stability of the protein, of the enzyme. And as the temperature increases, the kinetic energy of the particles and the structure increases, the vibrations within the protein will increase until there's a shape change. And with that shape change comes a loss of function. At the same time, we should consider the kinetic energy of the reactants, that is, the substrate and the enzyme. As their kinetic energy increases, we have increased kinetic energy. Therefore, they are moving more. Therefore, we have increased collisions, and that would lead to increased reaction and rate of reaction. These two separate ideas, one of a loss of stability and the other of increasing rate of reaction, resolve the two ideas to produce the classic curve, this asymmetrical curve of increasing rate of reaction at B and a rapid decline and loss of rate of reaction at D, which of course is known as denaturing, an irreversible change in the shape and the function of the enzyme. The peak of the curve corresponds to the maximum rate of reaction, and that occurs at a particular temperature, which is often referred to as the optimum temperature. In discussions, we perhaps mistakenly often believe that the optimum temperature is around 37 degrees, but this would only be true of ourselves. Other organisms have different optimum temperatures, and as previously mentioned, some of them are extreme, where in this Yellowstone National Park image, some of the bacteria in this lake have optimal pHs operating around about 70 and 80 degrees C. Extreme pH conditions within a cell also result in the same effect. If we have high pH, so basic conditions, or very low pH, acidic conditions, these can also result in a change in the shape, and this would also result in a loss of function. And we can see that in the two graphs below. However, A and B, two separate enzymes, have their optimum pH at different points. Nevertheless, becoming more acidic this way and becoming more basic this way does change the shape, which changes the function. It disrupts the structure of the protein, particularly things like hydrogen bonds are very sensitive to pH changes, and with the loss of the shape, the active site changes shape and the enzymes become denatured. In this instance where I've got A and B, the kind of idea here is that this would illustrate the optimum pH of the enzyme pepsin from the stomach, round about pH 2, as opposed to B, which could be salivary amylase, and that's optimal pH would be round about approximately 7. So very different optimums for two very different enzymes operating in two very different biological functional regions. The links below take us to further videos in the enzyme series, a button to subscribe, and a link back to the playlist for you to look at the other videos available in this series of Click for IB Biology.